Aha. Welcome to General Pharmacology. Let's move the PowerPoint slides and get started. We have plenty to cover today. Today we're going to talk about immunology, the study of the immune system. That's a hot topic right now with all the pandemic going around and around. And to understand immunology, the study of the immune system, then we have to study a little bit about blood. Look, this stuff gets ridiculously complicated. I took immunology at Baylor as an undergrad. Go Baylor. I took immunology as an undergrad with one of the pioneers. I wish I remembered his name, but he developed kidney transplant immunology, and he was our immunology teacher at Baylor. And we spent an entire semester taking that course. It's probably the most complicated course I've ever taken. I loved it. I loved my immunology class because it was so complex. And so miraculously beautiful on that matter. I turned around and took immunology again in medical school from one of our classmates in front of us, Craig Spellman. Why I remembered his name this morning is another miracle. But he taught immunology as well. So after 2 semesters of immunology, to pack this down into under an hour really doesn't give immunology the time that it deserves. So already I can tell you that if you're going to know anything about immunology, you need to know a little bit more than what we're going to cover today in our slides. And so one of the things with the clinical discussions is that the clinical discussions go into more detail about immunology. My slides cover so much basically for the television viewing audience. And then when you go to clinical discussion, those questions are going to ask you to further your understanding of these subjects. So that's one of the things that we do in clinical discussion, is give you the opportunity to expand your understanding of these things. And then there's this group project you guys turn it in to me at the end, and if it's really good, I'll take a careful look at it. Let's talk about immunity. I made these slides long ago, and so we'll talk about COVID maybe this semester. That is the plan. And we'll talk a little bit about COVID as we go along, since that's the disease that everyone's talking about right now. But to understand anything about COVID or any type of infectious disease and our body's response to it, we have to understand immunity. Immunity is about recognition, and if I can just connect 2 words in your mind today. Just remember that immunity is about the body's ability to recognize. And so your body can encounter some kind of infectious organism, whether it's a bacteria or fungus or virus, any pathogen, anything that can cause pathology. And this includes cancer cells in the body. I don't want to leave that out because we will talk about that later. So your body is able to recognize things that are a problem and launch a response to them. The first time it comes in contact with something, that's not really recognition. That's a part of the immune defense that's baseline. However, if you come in contact, when your immune system comes in contact with some kind of pathogen the next time around, then the body has developed this big response against that specific pathogen. So that ability to recognize and launch a bigger response each time, that's what immunity is. Immunity is the body's ability to recognize infectious disease-causing organisms like bacteria and viruses and protect the body from these organisms. And so we'll study hematology and immunology. We'll touch on microbiology. For as little as I talk about immunology, I talk even less about microbiology. And microbiology is a fascinating subject to get a very careful study in. Oh, and then epidemiology is the study of the causes, distribution, control of diseases in population. Epidemiology has really done more to combat infectious disease than anything else. Sanitation and hand washing and epidemiology have really done an unspoken amount of good in the fight against infectious disease-causing organisms on this planet. All right, this is a child with smallpox, and this child probably died. This picture was obviously taken back in the 70s because we don't even talk about smallpox anymore. Before 1979, smallpox killed 80% of the children who got the virus, and about half the adults. Since 1900, smallpox has caused 500 million deaths. Smallpox has been a scourge, a major killer of populations for centuries. And smallpox has been a very devastating illness. 67, that's right after I was born, 15 million got the disease, 2 million died. And smallpox was eradicated in 1979 through vaccinations. And so something in your clinical discussion section talks about the development of vaccinations for smallpox. And so I want you guys to take a look at that and see if you can research some of that story to see where the smallpox came from, or where the smallpox immunization came from, how that was developed, because that's going to be a completely different story than how the COVID vaccine was developed. However, the basic principles that make both of those systems work are what's in common in the body. Right now we're looking for a vaccine for HIV and AIDS. It's fascinating to me that we came up with a COVID vaccine as quickly as we did, and we're still on the lookout for an effective HIV vaccine. Now, HIV is a different virus than COVID, and how HIV hides from the immune system is different than how COVID hides from the immune system. So it makes sense that we're having a very difficult time finding a vaccine for HIV, and hopefully some of the technology that we've developed looking for a COVID vaccine will apply to the development of an HIV vaccine. However, before that HIV vaccine is developed, millions of people will die from AIDS. And so I've always said this, my goal is someday to teach this class and get an email from a concerned mom who says, well, why should I vaccinate my children for HIV? That disease has been gone for a long time. I don't want to vaccinate my kids for HIV. And then we'll talk about the risks and benefits. Smallpox has been gone so long that we don't vaccinate kids for smallpox since about 1979. So we'd like to eradicate HIV with a vaccine, and we'd like to completely eradicate COVID with a vaccine for that matter. All right, how vaccines work is to build up antibodies in your blood. These are called immunoglobulin antibodies, and immunoglobulins are the same thing. Now, when we think about antibodies, today we're going to talk about antibodies as part of the immune system. However, later we're going to talk about immunoglobulins that just quite simply function as drugs. If you remember Graves' disease, an immunoglobulin was stimulating the thyroid stimulating hormone receptor. The immunoglobulin was not attacking the thyroid. The immunoglobulin was actually acting as an agonist on that receptor. And so we can use immunoglobulins as drugs. And you'll notice that those monoclonal antibody, MAB, you'll notice that those drugs, those generic terms, end in MAB, monoclonal antibody, and we'll talk about what those are. And so when you see trastuzumab, any kind of generic term that ends in MAB, that stands for monoclonal antibody, and we're usually using it as a drug. And that's going to come later. But right now we're going to talk about the natural immunoglobulins that your body makes to fight with diseases. And we'll draw them as Y-shaped proteins. I have this very interesting animation that I'll share on our Lalima if you guys remind me if I forget. But it shows these little Y-shaped proteins going around and neutralizing the pathogens. And so this is a protein that we call an immunoglobulin, and we'll denote it as a Y-shape. And these are like receptors. When we first started pharmacology, we talked about keys and locks. And so you can think about these antibodies being like receptors, but they're not waiting for drug to cause some kind of regulatory change. They are on the lookout for antigens that might be pathogenic, or that it would recognize as pathogenic. And so when antigens are parts of the bacteria or the virus, specific proteins that will activate the immune system. In the case of COVID, these are the spike proteins, and we'll see that a lot when we start showing more pictures of viruses, more cartoons of viruses. We'll notice that the surface protein will be recognized by the immune system. So the surface antigens are on the virus. The antibody here is part of our natural defense, and it's like keys and locks. When a specific antigen meets with its specific antibody that's waiting for it, then it activates the immune system. Our definition of an antigen is any substance that causes the immune system to react to it. An antigen is any substance. I think we just said that. All right, and oh, no, we didn't. An antigen is any substance that causes the immune system to produce antibodies against it. All right, two different, very similar definitions of antigens. And so this is what we do in immunizations. With immunizations, we inject an antigen into the body that the immune system will respond to by making antibodies, so that when we actually come in contact with the actual pathogen, the actual bacteria, the virus that we've been immunized against, your body will recognize those surface proteins and have antibodies ready to stick against it and activate immunity against the pathogen. So antigens usually come from outside the body, but antigens may originate within the body. Sometimes your body has an immune response to itself, autoimmune disorders. That's what many of them are called. We're not going to talk about this too much, but I just wanted to make it very clear what antigens were. Antigens are anything that stimulate the immune system. And we're usually talking from outside the body when we talk about antigens. All right, so you have a wide array of these immunoglobulins that are all different types of receptors. They're on the lookout for different shapes and sizes of keys. And so different shapes and sizes of antigen. And so you probably have an immunoglobulin in your body that can stick to any antigen that's out there. And let's say that happens. Oops, nope, different slide. All right, and so here, what I'm trying to show you is that antibodies bind to a specific antigen. The reason I'm using the key is that only certain antigens with a certain charge and shape are going to be able to stick to a specific antibody. But once they stick, once the antigen meets its antibody, all this process goes on in the immune system so that your body can make more and more of these antibodies to be able to recognize the same antigen. This is where monoclonal antibodies, that word comes from. This has been a popular word to use in the treatment of COVID recently. They've been using IV monoclonal antibodies. Mono, one, clonal, from one clone. These are all identical copies of each other, nearly identical copies of each other, and they come from a single clone of lymphocyte. And so they're all identical and able to recognize the same antigen. So again, this is what we do with an immunization. And this is what your body does naturally. When it recognizes an antigen, it stimulates the immune system to make even more and more of the same antibodies to recognize the same antigen. And those will be called monoclonal antibodies. All right, so once the antibody binds to antigen, the immune system is activated. This is in your notes. I'll let you read this. Please do not confuse these terms, because when you get in a clinical setting, they start throwing these words around, especially when you're talking about testing. You're getting a test for an antibody. You're getting a test for an antigen. Is it a surface antigen? Is it a nuclear antigen? Is it a DNA antigen? Is it an antibody? And so this gets real confusing, so I want you to take some time to notice the difference between these abbreviations and these two terms, because like everything else, it'll melt together and you'll have a hard time. If you read carefully, there's two ends of an antibody. There's an Fc region that signals the immune system, FAB. is what binds to the antigen. I won't ask you that, but you might see this if you read carefully in immunology. All right, antibodies can be free in the blood, or all right, so here we have an antibody that's free in the blood, and it has come in contact with a specific antigen that it's able to stick to. And so that antigen is like a key that has activated this lock right here, and we'll call that an antigen-antibody complex. And what that'll do is cause a macrophage to recognize that antigen-antibody complex as needing to be digested. And so the macrophage will come and eat the antibody-antigen complex. And so not only are we consuming something made by the body, but notice already we're starting to fight with whatever this is. And so whatever that antigen is, that pathogen, whether it's a bacteria or a fungus or a virus, if an antibody can stick to it, then the immune system can start to respond to it and then cascade and build its response. All right, well, not all antibodies are free floating in the blood. Some antibodies are attached to lymphocytes. And so here is a B lymphocyte, and we can think of it as a memory cell. And so that B lymphocyte has these specific antibodies that are going to be specific for a specific antigen. And when it comes in contact with that antigen, that signals the B cell to make free-floating immunoglobulins into the serum. And so it'll make more and more of the same antibody, more immunoglobulins. And so again, this is how immunizations work. With immunizations, we usually give an antigen. We're doing something new now, but throughout the history of immunizations, what we've done is give the antigen to the person so the immune system can respond just like this. There's a B cell in your body that's going to be able to recognize that antigen, and when that B cell recognizes that antigen, not only does it make lots and lots of antibodies in the blood to respond to that antigen, but it also causes the B cells to reproduce so that they can keep making the same antibody. And so that's how we can use an antigen, a foreign antigen from a bacteria or a virus, and use that as an immunization to get the body to respond to that antigen with these various defense mechanisms. And so immunizations are just quite simply the antigens of disease-causing organs. What's new with COVID is instead of giving the antigens themselves, what we do is give the plans to the antigens in messenger RNA. And where is that slide? Let's go find it, because I want it on the recording. So we'll come back over here where my slides should be. Oh no, they're over here. There we go. This used to be in my immunology slides. So we'll go find the ribosome. Guys, remember any of this? All right. So what's new with immunizations? All right, well, antigens that we give as immunizations, they are proteins that are chains of amino acids that are put together. They're engineered to be the vaccine. In the old days, we used to use something called, and we still do this for some vaccines, something called attenuated viruses. We'll use virus parts. For a long time, we've used virus parts, that the virus was damaged, so it wasn't able to be a pathogen, but can still expose the body to the proteins. So what's new with the COVID vaccine is this right here. What we're doing, we're not giving the protein like we always have. What we're doing is giving messenger RNA. And what the messenger RNA does is tell our body how to code for this protein, and then this protein that our ribosomes manufacture will be the antigen that then the immune system responds to. So again, what we're doing with COVID is that we're giving the body messenger RNA. So our body's ribosomes will make this protein, and then this protein is what the immune system will develop its response to. And so it's not really too much different than what we've been doing all along. Messenger RNA technology as being able to be used as an immunization, we think that this is, the media would lead you to believe that this is all brand new. But if you read carefully about the history of messenger RNA, which I talked about using messenger RNA therapeutically, we talked about that the very moment the class began. And that technology has been around for a long time, and there's a very simple reason that it hasn't been at the forefront. Today, the National Institute of Health took half of the patent from Moderna. I think Moderna agreed to that. And that's a very interesting story about the history of messenger RNA vaccines. Just quite simply the transfer of intellectual property that occurred today because of that vaccine. And so, anyway, to make a long story even longer, the whole point is this. This is what we want. This is the result of the immunization. And so it doesn't matter if we give a weakened virus. It doesn't matter if we give a protein that's actually from a virus. It doesn't matter if we give a protein that's engineered in a lab, or if we give a messenger RNA that generates a protein that we naturally make that then the body responds to. The whole purpose of immunization is this right here, to have these huge number of antibodies that are specific to that disease-causing organism floating in the blood and ready to go against any pathogen that specific pathogen for which we've been immunized. And so the next time the body is exposed actually to the specific bacteria or virus, there's this memory that's built up against it. The body has seen this antigen, and then it's built up this response against the antigen. So the next time it sees the antigen in its natural form, it has this huge response that's ready to go. All right, so if we've been immunized against this bacteria, maybe the first time, if we've been immunized for this bacteria, then when we come in contact with this bacteria, there will be all sorts of antibodies that are able to recognize the bacteria and stick to it and signal the immune system for immediate destruction of that infectious organism. And so that's basically, in a nutshell, how immunizations work. All right, to make things more complicated, we have to study a little bit about blood. By the way, right now there are all sorts of great videos on how immunizations work. I like my version, but I'll see if I can remember to put some of these cool videos about immunizations in your resources section. I'll put them in chat, something like that, in announcements. All right, hematology is the study of blood. All right, remember what blood does? Yeah, it transports oxygen and carbon dioxide. What else does it do? Transport nutrients. What else does it do? Coagulate blood. And what are we going to talk about today? The blood's ability to combat infections. And so we think about the immune system basically being part of hematology, the blood system, because it's the blood cells that coordinate all this immune system response. All right, so let's talk about the parts of blood. Serum is the liquid base of blood. It's like salt water from the ocean. You can say there's tiny proteins in it, depending on your filter. We're going to talk about antibodies and complement. We've talked about antibodies as being immunoglobulins. Complement is similar to immunoglobulins. Complement is designed to randomly stick to things like bacteria. So complement is not super specific, and complement does not have memory. Your liver, when it makes complement, is designed to complement the immune system. And so when your liver makes complement and complement sticks to a bacteria and causes it to be digested, your liver doesn't remember that and make more complement for that. Your liver just keeps making complement. An antibody recognizes that bacteria and gets consumed, and that really signals the immune system response. All right, so complement's made by the liver to complement the job that the antibodies are doing. Antibodies are made of lymphocytes, a type of white blood cell. we'll talk about that. We'll talk about clotting factors. We did talk about clotting factors. You have red blood cells and platelets. We'll talk about that a little bit today. But what we want to talk about for the immune system is the white blood cells, the leukocytes. Leuko means white, cytes mean cells. That's why white blood cells are called leukocytes. And so we think of leukocytes as being the immune part of blood. The red blood cells carry oxygen around, the platelets help with blood clotting, and the white blood cells are about immunity. These are all made by the bone marrow, long bones, and in your pelvis bones as well. That's why if you get a bone marrow biopsy, they usually take that from the pelvic bones. All right, so this is a peripheral smear of some poikilocytosis here. All right, so these are some red blood cells and some platelets, and these are neutrophils right here. Neutrophils have many names: polymorphonuclear cells, PMNs, segmented neutrophils, segs, granulocytes. And so this is what's fun about clinical medicine, especially in infectious disease, when you're talking about infectious disease people and infectious disease experts. They'll throw these terms around to describe the neutrophils. They'll call them PMNs and segs and granulocytes. There are more granulocytes than just the neutrophils, but the neutrophils are the most common granulocyte. So when we talk about granulocytes, we're usually talking about neutrophils. And so polymorphonuclear, see how the nuclei have these different shapes? That's what polymorpho means. Segmented neutrophils, this is clearly a segmented neutrophil. We can argue whether this has become segmented or if it's still in its band form. But these are the neutrophils. They're the granulocytes. And there are other granulocytes besides the neutrophils, the eosinophils and the basophils. And so one of the stains is eosin, and the other stain is basic. And so the ones that take in the eosin are the eosinophils, and the ones that take up the basic dye are the basophils, and the ones that take up both, they're the neutrophils. And so that's where these words come from. And so eosin basically means red, and so eosinophils are granulocytes. You can see the granules in there. If you look carefully, you can see that's full of granules, and these little granules are vesicles that have antimicrobial properties. Same thing with an eosinophil. I photoshopped all this onto one slide. You shouldn't see this many white blood cells in one field. All right. There's a basophil. It takes up the basic dye. Well, you got to look under a microscope for a really long time to see a basophil. They're not very common. Less than 1 out of every 100 of these granulocytes is a basophil. About 1 out of every 10 is eosinophil. And so usually when you look under these slides manually, you'll see just sea of red blood cells, and you look, oh, there's a neutrophil, oh, there's another neutrophil. And you count to 10, 20 neutrophils you might come across in the eosinophil. You count to about 10 eosinophils you might come across a basophil. And then look right there, that's a lymphocyte, and see how it has that big cytoplasm? When that big cytoplasm is on the lymphocyte, that means the cytoplasm is busy doing something. And what it's busy doing in this slide is making immunoglobulins. And so you'll see this called a plasma cell because it's busy, it's been stimulated to make immunoglobulins. And so when you see a lymphocyte with a big cytoplasm like that, it must be making lots of antibodies. All right, so the granulocytes contain granules, and they carry all sorts of poisons designed to kill infectious organisms. That's a great read. What are inside the granules of the granulocytes? We could spend an entire semester talking about that. All right, but what I want you to know is that when you're working in the hospital, you're working in a clinical setting, you're going to meet people who have neutropenic precautions. Neutro as in neutrophil, penia as in too few. And so if they are on neutropenic precautions, that means they don't have enough granulocytes to fight infections, and so they'll be easily infected. And so somebody with neutropenia, maybe they can't have pets visit, maybe they can't have live plants, maybe you'll have to dress up and wear a gown to go into their room. There's all sorts of different levels of neutropenic precautions. And they're just quite simply called neutropenic because they have too few neutrophils, making them much easier to be infected. And so that's why people with a low white blood cell count are easily infected because we need our white blood cells to fight with infection, and granulocytes are very important. All right, so the eosinophils and the neutrophils and the basophils, those are the granulocytes. There are white blood cells that are not granulocytes, and those are the lymphocytes and the monocytes. The monocytes are the same thing as the macrophages that we talk about, Kupffer cells in the liver. There's all sorts of different strange types of monocytes that have different names as we go around to the different systems. Those are different than the lymphocytes. The lymphocytes actually coordinate the immune system, especially the T helper lymphocytes. And so these are two stars in the immune show that you will see. I don't have a picture of a monocyte, I don't think, do I? No. But that is the lymphocyte that we just got through talking about. I have cartoons of all of this. Hematopoiesis is how blood cells are made, the development of blood cells in the marrow. So everything we're going to talk about is going to be found on a bone marrow biopsy. The slides that we see in the blood are the ones we just got through talking about. We'll see granulocytes, we'll see some lymphocytes, we might come across a monocyte. But what we're going to talk about right now, we're only going to see in the bone marrow, because the bone marrow is like the nursery for the blood cells, and then at a certain point they'll be released into the blood system. If your body's having an infection, if your body's at war with some kind of pathogen, then your bone marrow will send its armies at a younger and younger age. We've seen that before in war. And so when there's an infection going on, your bone marrow will send younger, more immature white cells into the blood. And so when we see these immature white cells in the blood, that's another thing that tells us that there's infection, because we'll see these immature cells that are in the blood that we would usually only see in the bone marrow. And so we're getting ready to talk about all that. All right, when we talk about these two things, everyone gets really, really confused. And then when we talk about myelogenic cancers and lymphocytic cancers, no one has any idea what that means. And so we're going to make it simple right here. Okay, so can you name all the white blood cells? That is a great way to start. And the white blood cells are the lymphocytes. And then can you name all the blood cells, not just the white blood cells? Can you name all the cells in the blood? All right, well, one of them is the lymphocytes, and they can come from the lymphocytic line, the lymphoid line. And then there's all the other cells. The red blood cells, the platelets, all the white blood cells, except not the lymphocytes, are in the myeloid line. All right, so everything is in the myeloid line except the lymphocytes. The lymphocytes are in the lymphoid line. So if you can remember this, then you can remember all this too. Because I've heard somebody, name the myeloid cells. And they're like, and it's just like, well, everything but the lymphocytes. All right. And so it's really that simple. So if you can drill that into your head, you're going to be a lot more able to discuss things like myelocytic leukemias and lymphocytic leukemias and myeloid cancers and lymphoid cancers. Let's talk about the word differentiation. Differentiation is very simple. It just means to make different. As cells differentiate, they come closer to their final form. And so everything starts out as a stem cell, and that's the very beginning cell. And then that stem cell reproduces to make more copies of itself, but then that stem cell will reproduce, and as it reproduces, it changes. It becomes different. And as it continues to reproduce, it continues to differentiate until it's in its final form. And so when things are more differentiated, then they're more like the final product. And so later on, we're going to talk about cancer, and we're going to look under the microscope, and we're going to look at cells, and we're going to say how well differentiated they are. And if we say they're well differentiated, that means those cells look like their final form. If those cells are poorly differentiated, then those are cancers that look more like the original stem cells. And so we will use this differentiation term again when we talk about cancer and chemotherapy. But right now we're talking about the development of blood, and as these stem cells change, they are differentiating into their final forms. And so you can tell the cartoon pictures don't look any different. They look the same. We'd have to do special stains under the microscope to figure out which is the common myeloid and common lymphoid progenitor. And so, again, what are the myeloid cells going to become? Oh, all the blood cells except the lymphocytes. What does the lymphoid progenitor become? Lymphocytes. All right. All right, so here is the lymphoid line in one slide. All right. So again, it starts as a stem cell, becomes a lymphoid progenitor, and then that lymphoid progenitor becomes a lymphoblast. Blast is a cellular term that just means immature, very immature. And so a lymphoblast is about the most immature lymphocyte you can have that's still not a stem cell. All right. So now this is a lymphocyte, and it is a lymphoblast. So when we see that blast word, then we know it's in its most immature form. All right, something else that you should know about hematology, that pro word, that means the same thing as pre. All right, so that's a pre-lymphocyte. This is almost a lymphocyte. All right. And then from the prolymphocyte, it will continue to reproduce and make more prolymphocytes, but as it reproduces, it will also make cells that will differentiate into the main lymphocytes, which are the B and T lymphocytes. All right. There's other lymphocytes that are called natural killer cells. These are essential in fighting cancers. Certain drugs that fight with autoimmune diseases will interfere with these natural killer cells, and so that's why we talk about these. The B lymphocytes, we'll call these memory cells when they're waiting to be activated, and then when an antigen actually activates the B cell to start making lots and lots of immunoglobulins, then its cytoplasm will swell up and turn purple. This purple color is due to nucleic acids. And so when the cell is busy changing and reproducing, it uses lots of nucleic acids in its cytoplasm. And then as the cell becomes more mature, then it has less nucleic acids. It has the need for less nucleic acids in its cytoplasm. But the plasma cell is all full of messenger RNA because its ribosomes are busy making lots and lots of immunoglobulins. So plasma cells are an extension of the B lymphocytes. There should be a little arrow right there. That's the lymphocytic line. That's simple. All right, now let's talk about the myeloid line. When we talk about the myeloid line, the myeloid progenitor will differentiate into three different directions. The megakaryotes, the myelos, and the proerythros. All right, the proerythroblasts. All right, so erythro means red, and so a proerythroblast, this means this is going to become a red blood cell. All right. Anytime we talk about megakaryo, mega is big. Karyo is the cytoplasm. And so megakaryo, mega cytoplasm, we're talking about platelets. All right, platelets, they'll become this big, huge cell, and then they'll break up into the little platelet parts. And so that big, huge cell we call a megakaryoblast. And then the myeloblast will make the granulocytes and the monocytes. All right, so let's talk about the myeloblast. Here, my myeloid line starts with a stem, a myeloid progenitor, and those will differentiate, reproduce and differentiate, making copies of themselves, of course, and then making a myeloblast. And then the myeloblast will differentiate either into monocytes or granulocytes. And again, I don't expect you to memorize all these words. I want you to see the pattern, myeloblast. All right, this is the most immature a myelocytic cell can be. And then a monoblast, this is the most immature a monocyte can be. Promonocyte is a pre-monocyte, and then it's a monocyte. All right, so look at the monocyte line. It's blast, pro, and then monocyte. And we'll see this pattern. All right, well, I thought we talked about the neutrophils next, but next we have a drug for you to know about. All right, so again, here's the myeloid line. The myeloid line not only stimulates the monocytes, but the granulocytes. That's where we're going. And so that's what this drug, granulocyte colony stimulating factor, filgrastim, is Neupogen. It's also abbreviated GCSF, granulocyte colony stimulating factor. That's what GCSF stands for. It's one of the most common drugs that you'll give in the hospital. So many people who get cancer and chemotherapy and bone marrow suppression can become neutropenic. And one of the ways that we stimulate those white blood cells back in action is filgrastim, because filgrastim stimulates this step right here, the myeloblast going to the granulocytes, speeds that process up. But there's also granulocyte colony granulocyte macrophage colony stimulating factor, that is leuκine. Please do not confuse leuκine, granulocyte macrophage colony stimulating factor, with Neupogen, granulocyte colony stimulating factor, because I hear about people confusing these two all the time. And it's not a great big deal, but I wanted to make sure you guys knew the difference. And the reason that we go through all this is where granulocyte colony stimulating factor and granulocyte macrophage colony stimulating factor works on this myeloblast, helps the myeloid progenitor move towards the monocyte and the granulocyte direction. All right, let's talk about the neutrophils. Let's see, in the eosinophils and the basophils, we're going to talk about them all at the same time because each of them have the same names except for beginning with eosinophilic ones begin with the word eosinophil, basophilic ones begin with the word basophil, and the neutrophils, since they're most common, we don't say neutrophil before any of these words. And so here's my polysegmented neutrophil, polymorphonuclear cell. That's what these are called. And it starts out as a myeloblast, goes to a promyelocyte, myelocyte, metamyelocyte. Really what I want you to know from here is this thing called a band. You might come across a metamyelocyte, but these are the three most important cells that you can know from neutrophils. All right, so a neutrophil is a polysegmented neutrophil, a segmented neutrophil, a polymorphonuclear neutrophil is the most differentiated form. And so somebody who's healthy, who has no infection, it's pretty much all you're going to see under the microscope. When you come across a neutrophil, it's going to look like this. All right? But if you remember, if you go back and look at my picture, there was one that was kind of banded. And this is slightly more immature than this. And so when we look under the microscope and we see an elevated white count and an elevated granulocyte count, an absolute granulocyte count, and we look at it under the microscope, we see some of these bands under the microscope. That tells us the bone marrow is releasing these cells early in response to infection. And they'll call that a left shift. When we start seeing bands, and especially if we see metamyelocytes, then we're going to call that a left shift. Back in ancient times, when we lab technicians did this under the microscope, we had a counter. And the counter, as you went from neutrophil to band to metamyelocyte, that counter was off to the left. And so we used to call that a left shift. That's where a left shift came from. There was no such thing as a right shift throughout human history. Until recently, I was looking it up, and some very, very young lady was explaining what a white count with a right shift is. And so that's very interesting. That might be true. However, before she made that video, I've never heard of a white count with a right shift. And my understanding is the left shift came from shifting to the left on the counters as we used to do this by hand. And so I welcome any email that can tell me otherwise of why this is called a left shift. When we see bands and metamyelocytes under the microscope with an elevated white blood cell count, we'll say they have an elevated white count with a left shift, and that left shift means there's more neutrophils, and we're seeing some band shapes and even metamyelocyte shapes under the microscope. So that's what a left shift is. And you'll see the bands and the metamyelocytes for eosinophils and basophils too. It's just really hard to see the shape of a basophil nucleus under the microscope. And so we don't really talk about these. We never talk about basophilic bands. We don't talk about eosinophilic bands unless they have some kind of eosinophilic disease. They might talk about that. But neutrophils seeing bands on the microscope, that's something we call a left shift. All right, megakaryo, those are the platelets. And right before the platelets are formed, it forms a megakaryocyte. This is this giant cell. And then the cell just breaks apart, and the individual parts are platelets. And so this is where platelets come from. So I wanted you to be familiar with that megakaryo word. And notice it's just blast and pre, and then the cell. And so it's just that simple. If you can't remember, just think blast goes to pre, goes to final form. All right? And you'll be doing fine. All right, the red blood cell differentiation is very interesting. Red blood cells are just dumb bags of hemoglobin. They don't have to think. There's not really too much metabolism that goes on. So they don't need any of their nucleic acids. And what color do nucleic acids stain under the microscope? Blue. All right. And so erythrocyte is red under the microscope because it doesn't really need any nucleic acid. And so its nuclear material and genetic stuff, it just dissolves away and disappears, leaving you just with a bag of hemoglobin. So right before the erythrocyte is ready to go, it's a reticulocyte. And so the body will release, the bone marrow will release the erythrocytes into the blood while they're still in this reticulocyte phase. Well, then this brings us to a lab test called a reticulocyte count. And so what we do is we look under the microscope and we see how we have to stain the reticulocytes. Very special. We use a different stain for them. And after the stain, we'll look at the reticulocyte under the microscope and figure out what the percentage is of reticulocytes. If the percentage of reticulocytes is really high, that means the bone marrow is working really hard to replace the red blood cells because there's lots of babies in the blood. The reticulocytes are baby red blood cells. And so if we see lots of reticulocytes in the blood, we know that the bone marrow is making a lot of babies to replace those red blood cells. If we look under the microscope and see a very, very low reticulocyte count, that means your bone marrow is not working very hard to replace red blood cells. And so sometimes when somebody's anemic meaning they have a low red blood cell count, they have a low red blood cell count, we're not sure why. We might get a reticulocyte count to see if this is due to losing blood and your bone marrow is working really hard to replace it, or are you just not making blood cells? And so sometimes the reticulocyte count can tell us that. Before it's a reticulocyte, notice that it's cleared. It's a normoblast, has no color. It kind of looks like a lymphocyte under the microscope. And then pro and blast, and you know where all those words come from. All right, erythropoietin is a drug. Erythropoietin is a drug, but it's also originally synthesized by the kidneys to stimulate the production of red blood cells. And so this is why we see people in kidney failure that are anemic, because their kidneys are responsible for making a substance that stimulates the red blood cells. And this is also endocrinology of the kidneys. We don't really talk much about the endocrinology of the kidneys, but if you think about renin, it's control of blood pressure. Erythropoietin is another way to control blood pressure because the kidneys feel like the pressure's down, the volume's down. It can stimulate the bone marrow to make more blood cells. So erythropoietin can be given as a drug to stimulate the production of red blood cells. And this is commonly done when people have bone marrow suppression due to cancer chemotherapy. Their red blood cells will go down. They will become anemic. And we can give them this drug to help jumpstart their red blood cell factory to start producing more erythrocytes. But something to keep in mind about this drug is you have to be very careful about using this in people who are hypertensive. Why? Because erythropoietin is the kidney's way of saying, hey, I'm hypovolemic here. There's not enough blood volume. There's not enough pressure. Let's raise the pressure up. And so the kidneys deliver erythropoietin to the bloodstream to stimulate the blood to increase volume so it can increase pressure. And so erythropoietin, one of its main problems, one of its main unwanted effects is over time it can raise blood pressure because we're quite simply attempting to raise blood volume. So keep that in mind as you look at the package label for a Apoetin. Know the difference between the lymphoid line and the myeloid line. The lymphocytes are the lymphoid line. The everything else sites are the myeloid line. If you can remember that, it'll be easier to talk about cancers, cancer treatments. All right. Let's see, where are we? I think we have time to finish. I want to know how fast I got to go to wrap this up. Oh, I have a long way to go. All right. A long time ago, the immunology lecture used to start just like this. Matter of fact, these were some slides I had before pharmacology. All right. So every day, the human body is exposed to bacteria, funguses, virus, parasite, bacteria, viruses, fungi, parasites. There's a reason I use large font, and that's more than just so you can read this on your iPhones. Our body, on your Blackberries, I think that's a more likely way of describing when I did these. Our bodies are constantly fighting off these organisms. Your body is, we think about our immune system kind of like being the fire department, where there's a place where it sits and waits, and then when something happens, it gets up and scrambles, and it puts out the fire, and then it goes back to sleep. That's not how it works, and it probably doesn't work that way in the fire department either. Your body is constantly fighting infectious diseases. Infectious organisms, or your body's always fighting with antigens from outside the body. It's recognizing things and saying, okay, we have to respond to that. So that's always going on. So that's why you can have multiple immunizations at the same time, as long as you don't have allergic reactions to any of them, or any one of them cause a reaction problem. It's reactogenicity. But if you have a reasonable response to immunizations, you can give several at once. Why? Because your body does this all the time. It's what your body does, responding to these antigens, generating an immune response. All right, our bodies are designed to fight off organisms when they're encountered for the first time. But our bodies are able to remember many of these organisms, so we can fight them better for subsequent exposures. Now, some organisms, like the flu and COVID, they'll change over time. They'll change their surface antigens over time, and we won't be able to recognize them when they come back around. And so with the flu, we have to be re-immunized every year. With COVID, if some of the things that we're coming up with now don't work, we're probably going to have to be vaccinated against COVID every year or so. But some organisms, the part your immune system recognizes doesn't change. And so once your body's immune system recognizes that that pathogen is not able to invade the body and cause illness again. And again, immunity is the body's ability to remember these foreign substances, the foreign pathogens, and be able to attack them, respond against them even more vigorously the next time around. All right, so you'll hear people who say, oh, well, I'm immune to that drug. Well, you know, I took this drug for a while, and then it just quit working, so I'm immune to that drug. That is not how to use the word immunity. All right? Or somebody will say, oh, that bacteria doesn't work with that drug, and that drug doesn't work against that bacteria anymore. That bacteria has become immune to that drug. That is not what immunity is about. Immunity is about your body's encounter with an antigen, and then through the immune processes we're getting ready to talk about, amplifies the response against that antigen so it can remember it the next time around. All right, back when I had time to make fun cartoons, that's a virus. You can see its little attachment spikes on the outside. I drew this like over 10 years ago, and viruses haven't changed. And there's the genetic material on the inside. And here it enters a healthy cell and infects it. Well, something that's very common is that when a healthy cell is invaded, it will put the antigens on the surface. And part of cellular invasion leaves antigens on the surface. And so one of the things that the cell does is put viral antigens on the surface. And this helps the immune system know which viruses are infected. So instant replay, the viral antigens get left on the surface. The genetic material is taken into the nucleus to reproduce. The interesting thing about viruses, they can't reproduce on their own. Bacteria, you can put them in a petri dish and they'll split. They have all the genetic material and engineering they need, that if you put them in the right environment, they can just sit and reproduce, but viruses can't do that. Viruses cannot reproduce unless they enter a healthy cell and hijack its cellular machinery. And so here the virus has hijacked the cell and it put its genetic material into the cell's cellular machinery. So then the cell's normal cellular machinery will make all the parts that we need to reproduce the virus. We're going to talk about that next time when we talk about HIV and AIDS, because how the body responds, the mechanics of that are similar with a lot of viral illnesses, and that'll make sense as we go along. If you study immunology, they will divide it into cellular immunity and then humoral immunity. This is not like humor, like something's funny. Your humors are liquids that early in the Greek times, they referred to your blood as a humor. And so we can see this as blood immunity. And basically, humoral immunity just means the cellular signals that result in increasing antibodies in the blood. All right. And so there's not this clear black and white, well, this is cellular immunity, and that's humoral immunity. I'm like, well, there's mixing in the middle, like a Venn diagram that overlaps. There's overlapping between humoral and cellular immunity. But what I want you to understand about the difference between these two terms is their end result. The end result of humoral immunity is to generate more antibodies in the serum. That's the purpose of humoral immunity. And so when we're talking about immunizations, we're talking about increasing the body's humoral immunity against specific pathogens. However, that cannot be accomplished without cellular immunity. Cellular immunity are the cellular processes that basically have to occur for the end result to be not only humoral immunity, but an added layer of response. All right, so cellular immunity. There's a virus, and before the macrophage eats the virus, let's imagine that it's just this is the first time we've come in contact with this virus, and there are a few antibodies that are able to attach to this, and the liver makes complement that can attach to this. And so before too many of these cause infection, macrophage is able to get its hands on one because it's marked with complement and some random assortment of antibodies that are able to recognize it. Well, the macrophage eats the virus. Here's an electron micrograph that I stuck on here. I have an animation of this. This is even more cool. You can see that red blood cell back there. These little green things are supposed to be E. coli bacteria. So this is what a macrophage really looks like when it's reaching out and grabbing things and pulling it in. It looks like little tentacles. It looks like a little anemone. All right? It doesn't look like a PowerPoint thought bubble with a circle inside. All right? I used to have this used to work. Does it work today? That's what the warning sign was about. We'll find that some other time. This is a video showing how the little macrophages go out and attack something. Uh-oh. Is there something hiding there? Let's see. I guess not. All right. So again, after the macrophage digests the virus, just like when the virus entered a healthy cell, the macrophage will express viral antigen on its surface. And so when we talk about COVID and spike proteins is just a surface antigen on COVID. And once it has invaded a cell, that surface antigen will be exposed on the surface of the immune cells in combination with something called a major histocompatibility complex. This is a major word in immunology. If you don't learn anything else, just know that MHC stands for major histocompatibility complex. All right, anything that's a complex is this collection of proteins. It'll be like a city. And think about this. Maybe if you know, like if you look at a picture of New York City, can you tell the difference between a picture of New York City and a picture of the Wailuku? Well, of course you can. And so what a complex is basically this huge collection, this city of proteins that are kind of stuck together on this island on the phospholipid bilayer. And it's a way of the immune cells and the way for the body cells to communicate with the immune system. It'd be like a giant scoreboard at a game. And so there's a reason they call it complex. It is very complex, but it is a complex of proteins that signal cells about their immune status. So major, the most important, histo means tissue, and compatibility, how we get along. What's the most important way for our cells to recognize each other and recognize when they are sick is this thing called a major histocompatibility complex. And so if you do blood transfusions, if you do anything with any kind of transplants, the word major histocompatibility complex matching will come into your vocabulary. And so we're going to start that today. Major histocompatibility complex. And this is how immune cells identify each other and things that do not belong. And so the macrophage has taken viral antigen and expressed it as part of its major histocompatibility complex and releases something called interleukin-1. It's called interleukin-1 because that's the first thing we figured out how it worked. They're not numbered for any specific reason. I want you to go to the clinical discussion section. You can quit sending me emails and asking me if you have to do them. Just open the clinical questions and look at all the different interleukins that we use as drug. We'll call those cytokines as well. When we use interleukocytokines as drugs, they'll just be called cytokines. So I want you to be on the lookout for that word. Because they're interleukins, all right? And interleukins is interleukocytokine, and you'll see them called cytokines when they're used as drugs. Inter, in between, oh, there it is. Inter, in between, leuko, white, cytocell. Kine is kinetic, as in movements. And so interleukocytokines are how they communicate with each other and get them to move towards each other. And I had a video of the little blood cells moving around and chasing the pathogen. And the way they do that is with interleukocytokines. This is how cells are able to attract each other. And so interleukin attracts not only other macrophages, but attracts CD4 helper cells as well. Those are a type of lymphocytes. So again, the macrophage consumed the virus and it exposed viral antigen along with a major histocompatibility complex and signaled interleukin and released interleukin-1. And what does the interleukin do? Well, it causes more macrophages to come into the area and hopefully take an antigen and express it with this major histocompatibility complex. But something else that interleukin-1 does is signal our T helper cell. Our T helper cell is like the Superman, the mighty mouse. Here I come to save the day. T helper cell is on his way. All right, so I sang that this semester. T helper cell helps mediate all the cellular immunity. So when we talk about cellular immunity, the first thing that needs to come to mind is T helper cell. A T helper cell is the same thing as a CD4 lymphocyte. When we talk about HIV and AIDS, we're going to talk about CD4 lymphocytes. That's the same thing as a T helper cell. That'll abbreviate that TH. So interleukin-1 is called macrophage attracting cytokine, and it also attracts T helper cells. All right, so here's the T helper cell, and the T helper cell, it has an antibody surface on the T helper cell that can recognize this surface antigen. The surface antigen can be passed over to the T helper cell so it can express the surface antigen. Where is that? Oh, I guess I went backwards. Anyway, the T helper cell is signaled by, this T helper cell has been attracted and then it recognizes this antigen and signals interleukin-2, is able to recognize this antigen and release interleukin-2, and interleukin-2 just brings in more T helper cells. I'll let you guys take a look at that. By the way, when we talk about cytokines, we'll also talk about interferons, and interferons interfere with a viral ability to replicate its DNA and RNA. And so this is also going on with the cells, making these interferons. And so you'll see these clumped up together as drugs. But I wanted you to know the difference. Interleukins are how the cells communicate with each other. Interferons are how we interfere with viral DNA replication. All right, I think I'm missing a cell. But anyway, cellular immunity is this step right here. The T helper cell coordinating the rest of the system. All right, humoral immunity is about the B lymphocytes being exposed to antigen and then coming out of their memory state and starting to make tons and tons and tons of these antibodies. And the antibodies should be building up in the blood right now. It can expose itself to a free-floating antigen. The humoral immunity can also be stimulated by the antigen presentation by the macrophage, but the T helper cell can do cellular presentation or antigen presentation as well. Let's stop for a second here and take this slide and copy it and put that right there. Good. All right, so if you back up, remember our T helper cell got antigen presentation from the macrophage. One of the great things about the T helper cell is it can turn around and present antigen now to the B cells. So the T helper cells are able to take this antigen and present it on its surface and then attract B cells and stimulate them into making lots and lots and lots of antibodies. And these antibodies will stick to the specific antigen and mark them. They'll call it opsonize. When something's soaked in antibodies, that opsonization makes it really easy for the granulocytes and the monocytes to attack it. When we talk about antibodies, again, there's these Y-shaped proteins. We'll divide them into long chains and maybe short chains. And so people with some kind of monoclonal antibody disease, you'll hear it called light chain disease or heavy chain disease. And so sometimes if they make too many of these antibodies, we can see these light chains. So that's what I want you to know about that. Again, there's one side that the antigen binds to, and then the immune signal is on the other side. FAB is for antigen binding, FC is the immune signal. We talked about the difference between antibodies and antigens, so make sure you know the difference between these two terms. All right, so remember that virus. The first time we were exposed to the virus, your body started making all sorts of antibodies against it, and it got rid of it, got rid of the virus. Well, here the virus has shown up again. But what has happened is now there's lots of circulating antibodies waiting to mark the virus, and so it's very hard for the virus to get inside of the cell and reproduce once it's been opsonized, once it's been coated with these antibodies. And now that we've seen this virus the second time around, and the virus has not changed its antigen presentation, because some viruses do, but many viruses don't. And so this virus did not change its antigen, this is not able to change its antigen presentation. It's been opsonized and then easily metabolized. And so the first time we ate it, the first time the macrophage ate the virus, it was assorted, it had a random assortment of complement. But in this secondary response, there were these monoclonal antibodies that were waiting to attack that virus and mark it and ingest it into the immune system cells much quicker. We're going to see this when we talk about HIV and AIDS. There are different types of immunoglobulins. IgM is the initial immunoglobulin that is released when we're infected with something, and then IgG is like your memory antibody, and it will go up as well. All right, so IgM antibodies are called acute antibodies. It's very important in viral discussions that we talk about IgM levels and IgG. If we suspect somebody has a viral illness, we're not sure, we'll get IgM levels, and if those IgM levels are positive, they can tell us that we have acute disease. And so you might see this with the flu, you might see this with monocytosis. If IgM levels against Epstein-Barr are elevated, then that tells us that that's positive. The interesting thing about IgMs is they have this 5 segments. IgG is a single segment, single Y-shaped protein. It's called the chronotic immunoglobulin. This tells us if we're immune to something. So if you get a test to see if you're immune to something, they'll get an immunoglobulin test. It'll be an IgG test. All right. IgGs tell us we're immune to something. IgMs tell us we're being infected by something. Again, we're also going to use the IgGs as drugs later on. And so again, when we talk about antibodies, it's like, well, is it being used as part of the immune system and part of immunity recognition, or is it just quite simply being used as a drug to be an agonist on a certain receptor? We don't talk about this too much clinically, but there are the immunoglobulins that are on the surface of the B lymphocyte. Those are IgDs. IgAs are in what's in the fluid, bodily secretions, and so this is how breast milk translates immunity, passes immunity into the child through IgAs, and so all of our body fluids have these IgAs that protect us from pathogens entering our body. IgEs are immunoglobulins that are attached to mast cells that release histamine. And so if the person has some kind of allergic reaction, they might use IgE inhibitors. There's a drug in asthma that we don't talk about, but is an IgE inhibitor. It's a blocker. It prevents allergic reaction by blocking this right here. And so the allergic reaction immunoglobulins, those will be called IgEs. And you'll see some drugs that'll be anti-IgE, and they'll be used for fighting allergies and allergic types of reactions. The T lymphocytes, we divide them into CD4. Those are the helper lymphocytes. There's also the cytotoxic cells, the killer cells, CD8 cells. We'll talk about those when we talk about cancer and chemotherapy. Apoptosis is a word you need to know. Apoptosis means programmed cell death. So remember when the virus entered the healthy cell and made it frown? Well, we don't cure that cell. The cell does not get rid of the virus. What happens is the cell undergoes apoptosis, and that's through your major histocompatibility complex. The cytotoxic, the T killer cells, can go through and tell cells that their time is up, and the cells self-destruct and become recycled. Your cells don't last very long anyway. You're constantly making them. They wear out, they break down, your body carries them away and makes new ones. This is constantly going on. And so CD8s are important in slowing down all that immune response, as well as suppressor cells. All we talked about is amplifying the immune signal. There are also T cells that slow all that signal down when the reaction is no longer needed. We talked about B lymphocytes having plasma cells, which make the antibodies, the memory cells that are on guard for future exposure. Oh my gosh, and that's all I have for immunology today. Be sure and take a look at the clinical discussion for further information on what I was attempting to talk about today. When we come back, we're going to talk about HIV and AIDS, and then I think we might take a break in the schedule, spend a little time talking about what's new in COVID, since we actually have some people in our class working on that sort of thing. So until next time, aloha.